Welcome to the second part of a series of videos detailing how to perform ChIP-seq analysis using Galaxy. I'm going to start off by talking about how to actually get sequence into Galaxy itself. Then I'll be detailing a couple of programs used in the quality control of the raw reads that come from the sequencer. So let us begin by looking at how you can get sequences into Galaxy. Now, unlike the last time we looked at Galaxy, this is now our own instance of Galaxy at the University of Manchester. So it will look somewhat different to if you loaded up Galaxy main. So as before, we have all our tools or bits of software on the left hand panel. In the middle is how we interact with those programs and where you can look at results. And on the right hand side is a history, which in this case is the ChIP-seq analysis. The quality control step allows us to see whether the reads are suitable for downstream analysis. If there is a problem with the reads, we can then use trimming to remove poor quality nucleotides at the end of the reads, or to remove the adapter sequences that would prevent reads mapping. In our analysis, we begin with the raw sequence, and typically this will come to you as fastQ files. So here we are, this is the beginning of our Galaxy history and the history panel, and we have a ChIP fastQ file, which is the reads that are enriched for our transcription factor binding sites, and an input, which is the reads that have gone through the chromatin immunoprecipitation protocol but have not been enriched with the specific antibody. So this acts as a background control that we can use to compare against our ChIP sequences. So let's just make the history panel a bit wider by dragging this here. And if we want to see what information is contained within the history items, we can just click on its name. Because this is the first one, it doesn't look like anything's happened, but in fact it's appeared just below, so we can just pull this down. And if we click on the eye, we get a view of that file in the middle panel of the Galaxy tool. As you may have noticed, there is also the top few lines of each file in the history item, so you don't have to click on the eye every time you want to see what's in a file. So if we go ahead and click on that View Data Eye, in just a few moments that will appear in the middle. So here we have a fastQ file. The top line is the name of the read and the barcode here. Then we have the actual sequence, and then each of these corresponds to the quality for each base in the sequence. In this example, the reads are single-ended reads that are 68 bases long, although this is a bit unusual, as typically I'm dealing with paired end data that are 101 bases long. So you may be asking yourself, well, how do you get the files into Galaxy in the first place? And there are several ways of doing this. In the Get Data section, there is the upload file from your computer. You can either click on that, or we have this icon here, which goes to the same thing. So I'll click on that now. Now, in this section you can either drag files from your computer into this area and that will prepare them for upload. However, you can also choose to upload files from a web server with a URL, and you can do that here by clicking on the paste fetch data button. So if you have the URL for your web server, you can just type it in here. At the same time, you can also tell Galaxy what type of file it is, although it will try to guess what the file is by looking at the file itself. So in this case, if we wanted to upload a fastq file, we'd click on here and you can actually start typing, so fastq. However, you do need to select fastq Sanger for it to be recognised by downstream programs. Now at the moment, because this is unmapped data, we don't need to fill in what the species or genome is, so we can leave this as unspecified. If you're working for a university or a company, you might have easy access to a web server. However, if you're not so lucky, it's also possible to host files on other web servers such as Dropbox. There is a third option in that you can ask the administrator of the Galaxy instance to set up a data library for the information that you want to import, and this is accessible by going to the shared data area at the top of the screen and using the data libraries tab. There is actually a fourth way of getting data into Galaxy, and this is by uploading data to an FTP server and configuring Galaxy to import data files from that FTP server. With our ChIP fastq file and input fastq files loaded, we can now begin the quality control process using fastQC. Now, to find this in the Tools menu, we can use the Search Tools function because it can be quite hard to find the tool you want. So we can start typing fastQC, and we can see that it's installed under NGS QC manipulation. Incidentally, this is a tool that is available off of the public toolshed. So if we click on that, it will bring the tool interface into the middle panel. Before I carry on, here are some quick notes about fastQC. The version I'm using on our Galaxy instance is 0.11.2, and I recommend using any of the 0.11 versions as there were some significant improvements over the previous versions. The author's main webpage at the Babraham Institute is here. That's well worth looking at because there are links to their web manual, which has a lot of useful information. The author has also produced a YouTube video, which again is useful in determining what the different modules of the tool mean. One of the pieces of the results is quite tricky to decipher sometimes. There's a lot of information, and there's been a few posts about this on the internet. There was a blog post by Simon Andrews, the author, here, and somebody else independent of the project put an answer to the question about what the duplication plot means on the BioStars website. So the main aim of this tool is to identify any problems that there might be in the unmapped sequences, and this gives us an idea of whether to use the sequences at all, or whether there's anything we can do to save the sequences to make them usable in the downstream analysis. Before I start in earnest, here's just a couple of helpful pieces of information about fastQC. You can find the project web page at this link here, and Simon Andrews, the main author of the tool, has created a YouTube video which explains the major concept of using the tool. So the main aim of fastQC is to identify any problems in the unmapped sequence that we've got from the sequencer, and that will then allow us to make decisions on whether to use the data or not, or whether we can recover the data if there is a problem. The Galaxy tool page for fastQC also includes some interesting information about the purpose of the tool and the expected inputs and the type of output that is available. Running fastQC via Galaxy is very simple. All you need to do is choose the file or files from the top here. It will accept the fastQC files, which is the unmapped reads, but also will accept SAM and BAM files of mapped reads as well. And this can be useful if you have trimmed the reads, perhaps in the next step, and then want to look back to see what effect that has on the quality of the reads. However, for the moment, we're assuming that we only have the fastq files and haven't performed any further steps. You can run the analysis one file at a time, as in here. So we'd choose the first file, and then we could press execute. However, there's new functionality in Galaxy that allows you to run with multiple datasets at a time. So we could choose our ChIP fastq file, but also if we did control and clicked input, it would allow us to run the analysis in a so-called batch effect, so we could run both analyses at the same time. And that is all we need to run the analysis. So all you do is click execute and wait for the results. Then what you would see is these files appear, and when they're completed they turn green. To look at the results of fastQC, we should look at the web page version of the two sets of data. So these are the data for the chip.fastq file, and these are the results for the input.fastq file. Clicking on the history item in this case does not give us an awful lot of information. If we click on the eye icon, it allows us to view the HTML file that is produced. The summary section of the output file tells us whether individual tests have been successful, given us a warning, or have failed. However, it is important to note that just because a test has said to have been a failure does not mean that it cannot be used in downstream analysis. The basic statistics section is useful in that it tells us how many reads are in the sample, how long they are, and what version of the fastq file has been used. Modern sequences such as Illumina, ChIP-seq and MySeq sequencers will produce Illumina 1.9 or Sanger33 fastq files. For a comprehensive description of all the tests here, I would certainly go and look at the links that I showed earlier, as there is a very good manual and the video tutorial is very useful. Now what I'm going to do next is just go over a couple of the tests that I find particularly useful in deciding what to do with the data. I find that the most useful result to look at is the per base sequence quality module. This is a box plot that describes the quality scores of all the reads in the sample. The y-axis is a Phred score, P-H-R-E-D, that is the -10 log 10 of the p-value that the called base is not correct. So what we are looking for are very high scores, and that gives us good confidence that the sequence is real. So a Phred score of 30 would indicate that there is a 1 in 1000 chance that the base called is incorrect. In case you're unfamiliar with box plots, the yellow bar indicates the upper and lower quartiles of the distribution. The horizontal bar is the median. The blue line here is the mean value of the distribution, and the whiskers, which would be above and below, are the maximum and minimum values. As you can see here, the maximum value is at the top score of 40. We always tend to expect that the quality values will decrease over the length of the reads. However, sometimes the yellow bars can dip to a great extent near the end, and this can indicate problems with the sequencing. And in such a circumstance, trimming off the end positions of the reads can greatly improve the mapping of the reads down the line. Here is an example from the author's website that shows a set of poor sequences. So you can see that the Phred scores drop off at the end of the reads. However, as we will see in the trimming stage of the pipeline, it is possible to remove the reads that have the poor sequences, retaining a set of decent reads for the mapping step. The next result I want to mention often looks like there's a problem, but is just part of the Illumina sequencing results. So here we have the per base sequence content that shows the proportion of AC, T and G along the length of all the reads. You quite often see in the first 9 or 10 bases that there is a curious wiggle in the distribution, and this is because Illumina sequencing uses random hexamers to populate the flow cell prior to sequencing. However, these random hexamers are not truly random, giving this unusual distribution. So the next test that I find particularly useful is the sequence duplication level plot. Now what this plot tells us is whether the sequences in our sample of sequences are present only once or a few times. So the y-axis is the proportion of the reads, and the x-axis is the number of times that a sequence is present. So you'll notice that there are two lines in the figure. The blue line shows the total sequences in the sample and whether they are present once, twice, 3 times, etc. The red line shows the number of deduplicated, or what could more accurately be called distinct sequences. By distinct I mean the following. If there are 10 reads that are present once, then there are 10 distinct reads. If there are 5 sequences that are present twice, then that's 5 distinct reads. So in an ideal set of reads, and again this is from the author's website, we can see that most reads or most sequences in the sample are only present once and a few present a few times. The blue line indicates that there are fewer unique sequences than we'd like for a ChIP-seq analysis. However, there are also very few in the very highly replicated sequence categories of the graph. Now the effect of deduplication, or the distinct reads as I described before, looks better with more reads that are unique, but however there are still a reasonable proportion of non-unique sequences. Now this can be the result of there being very few binding regions on the genome. So this means that a large number of reads are clustering in the same areas, which reduces the number of unique reads. One of the problems of having high duplication levels is you end up with fewer reads in your final analysis, and this means lower coverage. So we can see at the top of this plot it says that percentage of sequences remaining if deduplicated, and you'd only be left with about 30%. Now for a mammalian genome, the recommended number of reads over a genome is about at least 20 million, although in practice 10 to 20 million is usually acceptable. The last 3 sections of the fastQC results page identify contamination with sequences that are not part of the genome you're trying to sequence. This includes the overrepresented sequences, adapter content and KMA content. And mostly what these will identify are contamination of the sequence with the adapters that are used to run the sequences on the machine. Generally what happens is that there will be pieces of DNA that are shorter than the run length of the machine. So for example, this sample was a 68 base pair run, so if there were pieces of sequence that were shorter than this, then the sequence would run into the end adapter. Now this is a problem for mapping, as it will not be recognised as genome sequence. But this is okay because the trimming step is able to remove this. So before I move on to the trimming step of the ChIP-seq analysis, I just wanted to look at the sequence duplication levels plot for the input.fastq, and this is interesting because compared to the chip.fastq, the level of duplication is a lot lower, and this is expected because the input DNA sample should be random DNA from across the genome, and the number of reads that remain after deduplication is also a lot higher, at 61% in this case. Just thought I'd mention a couple of little housekeeping tips for Galaxy. At the top of the history section you can see this cog icon, so if you click on that there's lots of different options for this particular section. If you remove history items using this cross icon, they can be retrieved again by unhiding hidden datasets. Also, if you have many history items open and you want to shut them all up to make it look tidy and you can see what all your history items are, you can click on the Collapse Expanded Datasets option, and they'll all close so you can see all the items clearly. The tool we use for trimming the reads for quality and adapters, etc., is Trimmomatic, and the version that I'm using in this tutorial is 0.32 and is available as a Galaxy tool from the public toolshed, and is actually one of the tools that we made in our facility. The authors have a good website that has a detailed manual, so you can reach that using this URL. So the main aims of this tool is to basically remove any adapters that are present in the sequence reads because the piece of sequence was too short compared to the length of the sequence run. And if adapters are left in the sequences, then they will not be mapped properly in the mapping step. The other thing we want to check is that the ends of the reads, which are generally going to be poorer quality than the start, are not so poor that they affect the quality of the mapping. And one final step is to ensure that no trimmed reads are too short for the mapping. I should just add that Trimmomatic has other functions for dealing with reads, but the ones that I'm using here work best for me when performing ChIP-seq analyses. The other thing that Trimmomatic does is that it is pair aware, in that it will use paired end reads and deal with them correctly. In that, if you have filtered reads from the forward and reverse ends of the pairs, sometimes you will delete one read's pair or partner read, and when it comes to mapping you need to have complete pairs. So Trimmomatic is able to deal with this and just output complete filtered pairs. However, in this case we are dealing with single end reads, so this is not a factor. Okay, so let's go ahead and run Trimmomatic. So we start off with our fastq files, which we have checked are OK by running fastQC, and I think it's important to mention that even though we didn't detect any particular problems with those sequences, I always run Trimmomatic just to make sure that the files are checked for any contamination with adapters and to check for the quality of the reads. The eagle-eyed among you may notice that this history has changed because I accidentally left out the trimming step. Let's go ahead and find Trimmomatic from our list of tools. Again, we'll use the search toolbar to find it. So Trimmomatic. Here we go, under NGS-QC and manipulation in this case. So we click on that. Okay, so here we have the interface for Trimmomatic. The first decision we have to make is whether our data is paired end or not, and in our case it is not, so we click on no. And this changes the interface for us, so all that we have to do is input our single fastq files. As before, we can use this multiple datasets input tab, which makes it simpler to run both analyses through one command. So in this case, we want to choose our chip and input fastq. Again, I've clicked once here and shift click to select the second. We can ignore these input trimmed and chip trimmed, because this will be the output of this exercise here. Okay, so we've selected our two fastq files. Now we want to select yes in perform initial Illumina clip step. This is the step that will remove adapter sequences. Now this first item here is quite important, as it is dependent on the sequencer that you used. So there's a few selections here, and basically you'll want to select ChIP-seq version 2 for any applications that used an older Illumina genome analyzer 2 or 2x, and that is the case for these samples. For more recent machines such as MySeq or HiSeq, you'd want to select the ChIP-seq 3. In some cases you may have used Nextera as the method, and this should be selected. So let's go ahead and choose ChIP-seq single-ended. So in general you can accept the default parameters, but you can always consult the manual to see what the effect of changing these has on the result. For the next step, we're going to accept this provided tool, which is sliding window trimming, and what this will do is it will move a short window of 4 nucleotides across every read, and when the mean base quality score drops below 20, it will cut the read. So what this does is it keeps the part of the good quality read and removes the poor quality end. To choose the last step of the process, I'll click on the Insert Trimmomatic Operation button, and I'm going to choose from this bar the drop reads below a specified length. And here I'm going to set this to 36 bases. So what this will do is, after a read has been potentially trimmed to remove an adapter, or trimmed to remove poor quality nucleotides, if the remaining length of sequence is below 36, it will just get removed. So this maintains a set of sequences that are at least 36 bases up to 68 bases. So at the end of this quality control process, we are reasonably happy that we have a good set of reads that are of good quality and do not contain sequence that will confuse the mapping step. Thank you for joining me.